All right, so let's look at a cartoon now of our ribosome. So here are the A, the P, and the E sites on the large subunit and the small subunit. Right now, you see the 30S and 50S. These numbers will be a little bit different depending on if you're talking about bacterial or eukaryotic ribosomes. Don't worry about that so much right now. But what I do want you to pay attention to is how the messenger RNA threads between the small and the large subunit. And you see basically the binding happening right here at the P site. So there are three stages to translation. All right, there's the initiation, elongation, and termination. There can be a fourth stage, and I'll let you figure that out because that's a part of the homework assignment this week. The bacterial and eukaryotic translation are similar. So let's talk about initiation, elongation, and termination. So in terms of initiation, there's also recognition, but I'll let you figure that one out. In initiation, we have the initial AUG start codon associating with the P site and the methionine-containing transfer RNA. So how does this happen? Basically, a messenger RNA comes into contact with a small subunit, and that attracts, it draws kind of like, I wouldn't say electrically, but it attracts the large subunit and they lock into place. And that's when the transfer RNAs can start interacting with the messenger RNA. As that occurs, there's also a second matching, like a microsecond later, maybe not even that long, happening at the A site. So this is the acceptor site, or I always thought of A for acceptor site. The tRNA comes in here. The peptide bond is formed as this amino acid gets transitioned, or I should say this amino acid gets transitioned over here so that this particular green ball, you would say, comes over here. And then you have termination occurring. Okay, so you have initiation, elongation, termination. So this happens when this cycle goes over and over and over again, adding more and more amino acids onto the amino acid chain. So here you have amino acid 1, 2, 3, 4. And as you can see over here, that's happened a few times. So you would be going through elongation, initiation. So elongation just goes back to more elongation if you have more codons to go. But once the stop codon is hit, then something called release factor lodges itself into the A site. What that does is it initiates the release of the completed protein, okay? So that can go float off in the cytoplasm and do what it needs to do. The small and the large subunits come apart, and you can see over here, this ... doesn't mean this was cut. That just means that this is a long segment of messenger RNA between the start and the stop codons. And at that point, amino acid 1, let's say this thing reassociates, initiates, I guess you could say, and you have the methionine sitting here at the P site, then the new methionine can come in. You have elongation, 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 elongation until you run into, or until the ribosome runs into the AUG stop codon, and then release occurs. Now that can happen a little bit differently depending upon if it's in, if you have release factors occurring in eukaryotes versus prokaryotes. So the messenger RNA, the initiator transfer RNA, ribosomal subunits all come together to form the initiation complex. We'll get to termination in a few minutes. Each of these require several factors or proteins, IF1, 2, and 3. They come together to help stabilize the ribosome. In bacteria, the initiator tRNA is F-formal methionine. Don't worry about that. It's still methionine. It's just a little bit modified. And this transfer RNA has been modified to this formal methionine. But again, you just keep thinking that it's a methionine. It's just a modified methionine. AUGs, in some cases in other organisms and bacteria, can be a GUG or UGG. For the most part, for this class, though, you can just think of AUG as the start codon. So here we're looking at upstream sequence. So I did mention that earlier on. What is going on? Well, the upstream sequence is really super important during initiation because it contains in it contains a sequence called the Shine-Delgarno sequence, okay, named after individuals. And so what happens is the 16S, or small subunit of the ribosome, identifies the start codon, but the Shine-Delgarno sequence is really important for also binding to this. Here's your small subunit binding to the messenger RNA very stably. So Shine-Delgarno is important for ribosomal small subunit binding onto the messenger RNA. And these are all hydrogen bonds, I want to point out. A, U, U, A, U. So, you know, the more hydrogen bonds you have, the stronger the link between the 16S ribosomal RNA or small subunit and the messenger RNA. So the Shine-Delgarno sequence is going to be located in this picture off to the left. IF3 and IF1 also help stabilize. And then IF2 binds to IF3, and it brings with it some GTP. You've probably heard of GTP before. It's kind of like ATP. It's just it does carry energy. It carries phosphates. But what it does is it basically sets everything up for the methionine to take place right here with the start sequence. Like I said, you have the small subunit, the methionine carrying transfer RNA, which is also called the Initiator Transfer Rna, and then your messenger RNA. After that, basically the large subunit comes in. It's kind of like a hamburger bun coming in on top. It finds its place, just guiding off of the methionine containing initiator transfer RNA, and boom, we're ready to go. So now transitioning to eukaryotes. All of this stuff that I was just talking about happens in prokaryotic cells. In eukaryotes, we have some other factors called eukaryotic initiation factors, EIFs. These guys are interesting because without them, it's difficult to assemble the whole ribosome with the transfer RNAs and the messenger RNA, because messenger RNAs are actually quite a bit bigger because we have, well, not all of them, but they can be bigger in eukaryotes. The initiator transfer RNA is designated here, tRNA MET, for methionine. So over here we have our initiation factors, IF1, 2, 3. We have elongation factors, EF2, TS, we just call it TU. And then over here in eukaryotic cells, we have eukaryotic factor 1 alpha and so forth. These are just different factors that are associated with initiation, elongation, and releasing of the protein after it's been produced. So the start codon is always going to be in eukaryotes and prokaryotes, the AUG, and we've talked about this a little bit. This AUG is what we call a consensus sequence. Basically, it's almost consistently start in almost every case. There are a few modifications, but don't worry about that. And so for translation to occur, we use what are called Kozak's rules after Marilyn Kozak. So over here, what Marilyn Kozak had proposed is that the start codon is usually the first AUG after the 5' cap, because AUG means methionine, and you can find methionine downstream as well, but it's that 5' cap that's going to make a difference. What are we talking about? Remember when we talked about messenger RNA having a 5' cap at the very front? Yeah, that's what's identifying the beginning of the messenger RNA. So if there's that 5' cap and then an AUG, that's when the messenger RNA, the methionine, can bind. It's really very consistent. So the messenger RNA is scanning for the right start codon, I guess you could say, and it finds it when it's close to the cap and the AUG. All right, elongation stage. In bacteria, elongation happens between 15 and 20 amino acids per second. In eukaryotes, 2 to 6 amino acids per second. So we're a little bit slower. Bacteria churn out proteins like crazy. So during this stage, amino acids are added one at a time. So it's this elongation step that we look at, where basically the ribosome is traveling along the length of the messenger RNA, reading the codons. So this one is where the P site is where you definitely have the appropriate transfer RNA in the right slot, and then the protein gets transitioned over here to the one that's right, right? And this is where the wobble can take place, is right here in the A site. After the tRNA goes to A, the old P that's uncharged now gets kicked out. So I always thought of the E site as the eject site, right? Here's the acceptor, here's the eject site, and here's the peptide bond formation, or the peptide site, I guess you could say. But the peptide bond is formed when this transitions over to the amino acid in the A site. So the uncharged transfer RNA is released from the E site. So the small ribosomal subunit plays a huge role in the codon, anti-codon recognition. In other words, it's a small ribosomal subunit that's doing the reading. It prevents the elongation until the right transfer RNA is in place. So if the wrong one comes in place, the wrong tRNA needs to be kicked out and the right one needs to come in. And here's what's amazing. There's only one mistake that occurs per about 10,000 amino acids. So for whatever reason, that 16S ribosomal RNA, okay, the small subunit, is the one that is really efficient at recognizing and grabbing the right tRNA. The peptidyl transferase is what transfers the protein, the growing peptide chain, onto the A site. So the 23S rRNA is the component of the 50S subunit that acts as the peptidyl transferase. I'm not going to ask you about this level of detail on the quiz. Don't worry about it. That's a different ribosomal RNA. Don't worry about it. And so the ribosome itself actually acts as an enzyme because, let's come back here, it's facilitating the transfer right here. It's catalyzing the transfer of this peptide chain onto the new amino acid. So this is one of those cases where the RNA is acting like an enzyme. So we call it a ribozyme. So this is the 1% of the time that an enzyme is not a protein. Remember, 99% of the times enzymes are proteins. So now moving on to the termination stage, the final stage occurs when we hit the stop codon, UAG, UAA, and UGA, and these all associate with release factors, RF1, 2, and 3, which identify the different sequences. Now, RF3 doesn't recognize any of those 3 codons. It binds to the GTP so that it brings it in to allow termination to occur, because you have to have energy to break the whole thing apart. So this is in bacteria, okay? In eukaryotes are always going to have these E in front of them. You have release factor 1 and 3. RF1, ERF1 recognizes the stop codons. 3 is also needed for termination. However, at this time, it's not clear why. So here's how release factor 1 or 2 in bacterial translation stops the process. And we've talked about this before. It comes into the A site, allowing the release of the protein and breaking apart the entire sequence. Interestingly, bacterial translation can begin before transcription is completed. In other words, as the messenger RNA is being made, it can be translated immediately, because in bacteria, there is no nuclear envelope separating the ribosomes from messenger RNA. So you can have coupled transcription and translation. So here are the different types of ribosomal subunits, the different sizes. Initiator Transfer Rnas, the Shine-Dalgarno sequence, and so forth. This is probably going to be an important figure or table to look at for the exam. Now, how do you stop translation? Well, if you want to stop translation in bacteria, let's say to, stop you from getting sick, you can use these compounds that we call antibiotics. Say, for example, tetracycline. Tetracycline blocks elongation by inhibiting tRNAs, the aminoacetyl tRNAs, from going into the ribosome. In other words, it doesn't allow the charged transfer RNAs to go into the ribosome. Therefore, bacteria can't make new protein, they die. Streptomycin interferes with the binding of the tRNAs with the codons. These two we can use in humans to some degree. Erythromycin, chloramphenicol completely blocks the formation of the small and the large subunits.